Hey guys, today we're at Bowling Green State University looking at BG's own laboratory for sensory ecology. The lab is overrun by Dr. Paul Moore, and about 18 years ago is when the lab was established. Since then, it has been home to hundreds of students, both undergraduates and graduate students, who are interested in trying to understand how different organisms on this earth perceive their environment. Let's go check it out. All right, so now we're actually in the lab for sensory ecology, and with me I have Amanda, and she's actually one of the grad students that works in the lab here. So Amanda, can you tell us a little bit about yourself and your research?
Sure, so I'm in the second year of my master's, so it's my final year. My research focuses on sublethal levels of an herbicide called 2,4-dichlorophonoxyacetic acid. It's most commonly known as 2,4-D, so that's what I'll refer to it as. But I was looking at the levels, the sublethal levels, and how they impact crayfish in their foraging behavior, so their ability to find food. So this is a rusty crayfish. They're actually an invasive in our area, but unfortunately they've almost completely replaced the native, which is virulus. So these are what most of our studies are using, just because they're the ones that we find most commonly in the Portage River, which is right down the road. So why crayfish are cool to use for sensory experiments is because they have these really long antennae and antennules that are full of these chemoreceptors. So chemoreceptors can detect chemical stimuli and signals in the environment, so they can detect predators, they can detect conspecifics, so members of their same species, they can find food. So many different things can be perceived through using these chemoreceptors on the antennae and antennules. Cool.
Wow, very interesting. All right, well, thank you very much for your time. I really appreciate it.
Thanks for talking with me.
Not a problem. Hey guys, so now we're outside of the laboratory for sensory ecology, and with me I have Sarah. She is actually a student at Bowling Green going for her PhD. So Sarah, thank you for meeting with us.
Thank you for having me.
What can you tell us about yourself and what you're studying here?
I am a third-year PhD student, so I have about a year and a half left of my degree, and my dissertation research is focusing on the effects of sensory pollution and the types of information that are available to animals. So specifically, I'm looking at how chemical pollution impacts the crayfish's ability to perform chemically mediated behaviors.
Okay, such as?
Such as orientation behavior.
Okay, how did you form your experiment in order to test?
So I actually worked as an environmental consultant for a few years between my master's and PhD work, and I worked specifically as a water quality specialist. So I was looking at the types of chemicals that were occurring in ecosystems, and they were above levels that they should be. So I really became interested in how those levels were going to affect the aquatic ecosystem. Very
cool. Well, good luck with your PhD work and getting approved for that. And thank you very much for meeting with us. Thanks. I really appreciate it. So as you guys can see, there's lots of different ways in which BG students and faculty are working together to contribute to the science world to understand how different animals perceive their environments. From the Laboratory of Sensory Ecology, I'm Brittany, and I'll see you on my next wild adventure.